Well, first of all, I'd also like to thank Mary Smart and William Smart for funding this symposium series. I think it's really important that people in science and chemistry have the opportunity to interact with students and to educate the public about the contributions of science. And this is a great honor for me to be part of this, and I really appreciate this opportunity. I'm also grateful to be able to come back to Pensacola because, like Mike said, I came here as an undergraduate. I was raised mainly in St. Petersburg, but I spent every summer on a dairy farm in Wisconsin. My relatives were farmers, and my grandfather really wanted me to take over the family farm. And my father, who had put himself through school on the GI Bill after World War II, really wanted me to go to college. And so eventually I decided that if I didn't go to college now, I never would. I was actually up in Wisconsin, and I had a friend register me at St. Petersburg Junior College. I said, all right, I'll go back and I'll take some classes now, but I thought I really wanted to farm. And at St. Petersburg Junior College, I was inspired by an organic chemistry teacher named Nina Milton. And this was a wonderful woman who was really tough, but she really cared about her students. She had really high expectations, but she actually, when she saw that some of her class was interested in possibly majoring in chemistry, she drove the entire class from St. Petersburg to Pensacola because she felt that this was the best place to get a degree in chemistry. And so this was a whole weekend event, that she took out of her life for an organic chemistry teacher, and I came up and had the opportunity to be really inspired, even at a greater level, by the faculty at West Florida. So Jerry Gerst is back there. He was my organic teacher. Peter Tanner is in the front. Who else from West Florida is here who was on the faculty when I was here? I think it was just the two of you. And I found the same kind of environment at West Florida, where Jerry Gerst actually drove us to New Orleans into Atlanta so that we could visit the graduate programs. And you really have a great resource here, and maybe you didn't know it, but it's a wonderful role model, I think, for how academics should be run. The faculty there taught me not only how to think like a scientist, but also how to be a mentor. And so what I'm hoping to do today is show you how I have, in a way, followed in their footsteps, first of all, by trying to do some interesting science, and second of all, by trying to take some of the creative approaches we have in science and applying them to mentoring and to education. And we've been focusing on a group of students that you normally don't think of when you think of high-power science careers. These are typically underrepresented minority students and women that you often don't see attaining high positions in the sciences. So I'm going to take a little bit of time at the end of my talk to tell you about our efforts in those areas, because I think you'll find them interesting, and I think it's fitting given the nature of this award, this talk. So I want to give you a disclaimer up front. I'm not a clinician. I don't have a medical degree. I'm an investigator with the Howard Hughes Medical Institute, but that is a program that funds mainly scientists like me. It does fund clinicians as well, but most of the people are scientists like me who are trying to understand how things work in biology. And so I'm going to show you how I took my training as a chemist and tried to use it to understand how the AIDS virus works. And based on that information, we hope that someday we will be able to develop drugs that can combat highly resistant strains of HIV that are now beginning to emerge. So let me give you a little bit of background about the virus. This is from a 2007 United Nations report. There's a more recent 2009 report. I just didn't get the numbers into the slide, but there have been nearly 30 million deaths due to AIDS since 1981, when the virus was first discovered. There are currently, this number is still actually about the same, there are approximately 40 million people who are living with HIV, and about half of those worldwide are women. And in any given year, and this is the same for the last year that the data are available from the United Nations, so in 2010, there were about 3 million deaths worldwide due to AIDS. There were about 4 million new people who became infected. And what this translates to is that in any given day, so today, about 14,000 people became infected with the virus. Now, you hear that the problem is acute in third world countries and sub-Saharan Africa, but actually in the United States, in most major cities, the problem is just as bad as it is in sub-Saharan Africa. So in the United States, minorities are disproportionately affected. I do a lot of work with African Americans, and African Americans make up about 12% of the US population. But in the United States, they actually make up about half of all HIV cases. And in our nation's capital, which is only about 30 miles south of where I live, 1 out of 20 residents are HIV positive. So it's still a big problem, even right here in the United States. So what does the virus look like? I mean, if you wanted to try to develop a drug, you really want to kind of know what does it look like. And the problem is that viruses are very small. And with the most powerful electron microscopes, we can see what the virus looks like as it's budding out from a cell. So this is a cell that has been infected with the virus, and the proteins of the virus are assembling at this membrane, the outer membrane of the cell. And so you can see this is starting to form, and then eventually this will pinch off, and you get a virus particle. And this particle can then go on and infect other cells. Now, in order to develop a drug, we need better resolution than this. We can't just look at a fuzzy-looking image like this and try to develop a drug. We need to know what the three-dimensional shapes of the molecules are that give rise to this fuzzy-looking structure. And the problem is that microscopes are not powerful enough to get resolution that's needed. We need to be able to peer down inside this fuzzy stuff and see what the molecules look like. And because I was trained as a chemist, that's what I wanted to do. So what do the parts look like? Now, I use a technique called NMR. So everybody here has heard of MRI, right? So NMR is like MRI. NMR is, the acronym is for nuclear magnetic resonance. And NMR has been around for a long time, way before MRI. But when NMR people like me, when I was at the NIH, started developing this new method for imaging people, they decided, well, we better not call it nuclear magnetic resonance imaging because no one will want to get near the thing. So they dropped the nuclear, but it's the same technique. And basically it involves putting something inside a very strong magnet and trying to see then what the structures look like. So with MRI, we can look at big structures like muscles and bones and tumors and things like that. With nuclear magnetic resonance, we can look at very small things like molecules. And so we're using this technique, nuclear magnetic resonance. Now, for MRI, the magnets they use are very strong, so you wouldn't want to go into an MRI machine with a pacemaker or with a credit card, right, that you wanted to use later, or a watch. But the machines that we use are even stronger. The magnets are so powerful that if you can go on YouTube if you want and watch people have lowered frogs down into the middle of the magnet where we put our samples, and they levitate just due to the residual magnetism in their bodies. So super strong magnets. You wouldn't be able to do MRI in that kind of that strong of a magnet. But that's what we use. Now the magnet that my laboratory uses 12 years ago was the most powerful magnet made. The NIH bought it for us. And at the time it was being installed, I was actually here giving a talk at the University of West Florida. And so my wife was on the top of the library videotaping them installing this magnet. And it was an unusual installation because there was no way to get it into the courtyard. So the way they did it was they dangled the magnet. Here's the magnet, and it's being dangled from a Sikorsky helicopter up here. And this is how they were moving it into the building. Now the helicopter was rated to carry 10,000 pounds. And the magnet weighed 9,500 pounds. And it was a windy day. And so what they did is they had to evacuate the biology building, and they took all the extra fuel out of the helicopter and all the extra parts because they said, look, if we get a real serious downdraft, we're dropping it. And that thing weighs so much, it would have gone right through the building. So anyway, they got it safely in place, and that's the magnet that we still use today. Here's a picture of it installed. This is a large, what it really is a big thermos jug. And it's filled with liquid helium and liquid nitrogen. And the liquid helium is so cold that the wires that are used to make the electromagnet are superconducting at that temperature. And so we have a very strong magnet. So about 15 years ago, when this was installed, the engineers hooked up a power supply, got the current flowing in the magnet, disconnected the power supply, and the current's been flowing ever since. And it'll never stop flowing as long as we keep the magnet cold, because there's no resistance in the wire. And that's how we can get those magnets so strong that we can use the techniques that we're talking about. And actually, there's a hollow bore that goes all the way from the top of the magnet down through the middle, and the most powerful part of the magnet is right in here. And so we use cushions of air to lower our samples down inside, and this is where that frog would be flopping around. But our students walk up the steps and then lower the samples down in, and this is where we collect data for our samples. Now, what I'm showing is a cartoon of the AIDS virus. And this is just an artist rendition. The virus has a membrane on the outside. And then these cartoon drawings are protein molecules that actually have different functions. So there are proteins on the surface. There are proteins inside the virus. Some of these are color green. Other proteins that assemble into this coffin-like structure in the middle of the virus. It's been called the warhead of the virus. This contains the information that's needed to infect a new cell. So there are lots of proteins inside this warhead, and there's nucleic acid. So we all know that we have DNA as our genetic material. This virus, it's a retrovirus, and that means that it uses RNA as its genetic material. And all this virus needs to do is get inside a new cell and get this RNA inside a new cell, and the cell will then use that RNA to make lots and lots, hundreds of thousands of new viruses, and they go in and basically kill the immune system. Now, what I'm showing around here is 10 years of efforts to figure out what the three-dimensional structures of these molecules really look like. And so when you see a drawing like this, this is a way of representing the folding of the protein that's on the surface. And somebody who does drug design might be able to look at this folding and try to develop an inhibitor of this molecule. And I'll talk a little more about that later. To get this kind of high-resolution information about the folding of a protein, again, we can't use microscopes. And so there are two general ways of doing this: X-ray crystallography and NMR spectroscopy. And NMR played a really big role in the first decade of HIV structural biology. Actually, these 7 proteins were solved originally by the technique that I'm going to be telling you about, NMR. And the 3 major structural proteins of the virus, so one of them is called the matrix protein, and this is the one I'm really going to be focusing on in my talk today. This protein is in the mature virus. There are many of them, and they're stuck on the membrane. And this was solved in my lab. There's another protein that is stuck on the RNA, and one that helps form this coffin structure, and all 3 of those were solved in my lab. And actually, what I'm really proud of is that they were all solved with real heavy participation of undergraduates and minority students. So, we have a general idea of how they look, but how do those different parts of the virus function? So the virus is, in a way, it's like an engine, like an automobile engine, only it's a lot simpler. An automobile engine, I don't know how many thousand parts there are that all have to work together. It's got spark plugs and a crankshaft, and everything has to work right. Well, the virus has these different parts as well, and they also have to function in a certain way. They have to interact with each other, or sometimes they have to interact with the cell. And so if we understand how they work, then we might be able to understand how to inhibit them. Just in the same way, if you understand how an automobile engine works, then you would know if you pull off the spark plug cables, the engine won't run. So it's kind of the same idea with trying to develop an inhibitor to HIV. So what I'm going to do is give you a kind of an overview of how the virus works, and then I'll focus in on the protein that we've been studying. So this is that same cartoon of the virus. And the immune cells have their own proteins on the surface, and these are what are called chemokine receptors. And what they do is they will bind, they just stick to these proteins on the surface of the virus. So other cells don't have these, and so if the virus bumps into a different cell, it doesn't really stick to them, or if it does, nothing happens. But if it bumps into many of the cells of the immune system, it sticks to those cells because of these protein-protein interactions. And then what happens is the membrane of the virus fuses with the membrane of the cell, and the contents of the virus get inside this human cell. Now what happens is the virus, remember I told you it contains RNA as its genetic material. There's a protein that comes with the virus, and it has a name you've probably heard of, reverse transcriptase. You may have heard of reverse transcriptase inhibitors. It's one of the very popular inhibitors of the virus. This is a protein that copies the viral RNA into DNA so that now it's compatible with our own genetic material, the DNA of the cell. So like I already said, one of the first classes of inhibitors actually interferes with this enzyme and blocks this step of the virus life cycle. Those inhibitors work reasonably well, but by themselves, they're not sufficient to actually completely treat somebody. We need inhibitors to other parts of the replication cycle as well. So in the absence of those inhibitors, the viral RNA gets copied into viral DNA, and the DNA then gets taken into the nucleus of the cell and actually gets inserted into the DNA of the cell. So now the cell's own DNA actually includes the viral DNA. And this actually has gone on throughout evolution. The human genome contains a very large proportion of DNA sequences that came from old viral infections. They're called human endogenous retrovirus elements. And so this kind of thing has been going on throughout biology. In this case, though, in the case of those other infections, the cell has come up with ways of coping for them, and so they no longer lead to disease as far as we know. In fact, they've probably been beneficial to humans in the long run. In this case, if we have the HIV DNA in here, it creates a real problem, because the cell then normally reads DNA and uses the DNA to make cellular proteins. Now, what happens is the cell reads this DNA and it doesn't know that it's a viral DNA, and it just does what the DNA says. It says make proteins. And this is a cartoon drawing of the viral protein that gets made. Now, there's only one acronym that you kind of have to, or two, I guess. This protein has been called GAG, G-A-G, its historical name, and all retroviruses, including retroviruses that cause leukemia and cancers, other cancers, make this GAG protein. Now, they're all different in subtle different ways, but they all have these main features. There is a part of the protein at this end, and it's called the matrix domain, and this is the end I'm really going to be talking about. And then there are other parts that all have different functions. Once this protein is made, many copies of this protein have to get to the surface of the cell so that they can assemble into a new virus. So thousands of copies of this protein will actually somehow get to the assembly sites on the membrane of the cell, where they will bud out, just like I showed you in that electron micrograph in the first slide. They will bud out to form a new particle. Now, this particle is non-infectious. What has to happen now is there's another enzyme in the virus called protease, and you may have heard of protease inhibitors. They're another very popular, widely used inhibitor. The protease actually inhibits the enzyme that has to cut this big protein into pieces, and these color-coded domains then rearrange to form the virus, the infectious form of the virus that can go on now and infect a new cell. So again, the two main mechanisms that we have for inhibiting the virus are a group of drugs that bind to the reverse transcriptase, and block this step of the replication cycle, but by themselves they're not quite good enough. And then there are a group of drugs that inhibit the enzyme that leads to maturation of the virus. And again, by themselves, this group of enzymes really isn't good enough. But when these are used in combination therapies, the virus replicates at such a low rate that it's not able to evolve around the drugs. So if everybody who is HIV positive could be treated with the drugs that are available today, and they were compliant and they took those drugs, HIV wouldn't really be a big problem. People probably would live normal lifespans on the existing drugs that we have. But here there are two real big problems, and I'm sure you know about them. Number one is that the drugs are expensive, and there are a lot of people in third world countries who don't have access or don't have the money or just can't physically get to the drugs who are HIV positive. But in the US, there's a big problem as well. Some of the people who are HIV positive also have other problems. Some are IV drug users. Some are in the sex trade. Some have other issues. Some of them are just not wealthy enough to be able to afford the drugs. Some people may have enough money, but they would use the money. First, they start taking the drug, and they feel better, but then they want to feed their children, and so they use the money for other things. And the real problem in the US is one of compliance, where somebody who is HIV positive, they go on the drug, then they don't take it in a regular way for whatever reason. And when that happens, the virus rapidly mutates and evolves. And it now becomes resistant to those drug cocktails. And there was an article in Science magazine about 6 months ago that predicts that just based on data in San Francisco, the US could be set for an outbreak, perhaps of epidemic proportions, of this highly resistant, one of these highly resistant strains of HIV. So even though it might seem like we've solved this problem, at least to some level, we really haven't. We've got to keep working to try to be ready for these highly resistant strains of the virus. Now, what would you do if there was a highly resistant strain? How would you tackle it? Well, one way to tackle it would be to try to come up with drugs that target different parts of the virus life cycle that haven't already been targeted by the current drugs. And that's really what we've been trying to do. And the part that we're focusing on is this part of the virus, the matrix domain, because we now know what this function of this domain is. This domain of the protein, its job is to get the protein to these assembly sites. And it does so by requiring a little tail on the end of the protein, and for the chemists in the audience, this is a myristic acid. But basically, it's a tail on the protein, and a lot of cellular proteins have these similar tails, and what they do is they are strong anchors to bind to a membrane. And so the virus has one of these, the protein has one of these tails that anchors the protein to the membrane. But we want to know how do the proteins know to get to the right membrane, because this is the plasma membrane, the cellular outer membrane, where the virus has to bud. But there are lots of other membranes inside the cell. Just shown on this diagram is the nuclear membrane, and there are other membranes I'll show you later. How does it know to get to this right membrane? This structure doesn't tell us that. So several years ago, almost 10 years ago now, my lab solved the three-dimensional structure of the matrix domain with its tail on there. It's this myristic acid. That myristic acid now is shown in red, and the rest of the protein is shown in this fashion where you can sort of follow the backbone atoms. This is just a long chain, and it has these different folds. The circular curlicue kind of things are designed to denote something that's helical in structure. And when you see these arrowhead-shaped structures like here, that's designed to denote a different kind of folding of the protein called beta hairpins. So anyway, this tail, what you can see is it's tucked down inside the protein. That was a real puzzle for us, because the tail, this is supposed to be a membrane anchor, and this is not going to be able to anchor the protein to a membrane if it's stuck down inside the protein. So how can this function as a membrane anchor in this sort of structure? Now, virologists from around the world had already shown that this tail is critical for the function of the virus. Again, it's a meristal group, and virologists had shown that if you modify the cell or the matrix domain itself so it doesn't get this tail put on, the virus is dead. So it has to have this tail in order to work, in order for the virus to assemble. But as I said, it's sequestered inside the protein. So the first question is, how can this meristate anchor the protein to a membrane if it's buried inside? And there were other puzzles at the time as well. This is an image of a cell showing the whole cell. And in this case, those viral proteins are color-coded green. And what you can see is that the proteins are assembling at discrete sites on the plasma membrane. They're not just assembling anywhere. So these punctate sites serve as the sites of assembly. So, a couple of questions. First, what attracts the proteins to the plasma membrane, this outer membrane? And then second of all, what attracts the proteins to these punctate sites? So these are all questions that nobody had any idea what was going on, and we thought we needed to know how they're getting to these sites to assemble in order to design a drug that might block that process. So the first real keys to try to figuring out how the viral proteins get to these assembly sites came from work that was done at the National Cancer Institute. And what that work showed is that the virus hijacks a chemical signaling system inside the cell. So for the chemists that are here, here's the chemistry. This is a molecule that is found in different membranes inside a normal human cell. So here's the plasma membrane, and these molecules are in the plasma membrane, they're in the nuclear membrane, they're in membranes of a structure called the endoplasmic reticulum, a structure is called Golgi, lyso. These are all different structures that are inside a normal cell, and they all have membranes. Now, cellular proteins have to get to specific membranes as well. So how do cellular proteins get where they need to go? Well, cellular proteins have been designed, many of them have been designed to recognize these molecules. And these molecules are slightly modified in different membranes. So in the plasma membrane, the molecule is modified at the 4 and the 5 position to have what's called a phosphate. It's a phosphorus atom at this spot. But in other membranes, it has phosphorus atoms at different positions. And so a cellular protein that has to get to this outer membrane sometimes will have a site on it that will recognize this molecule and not recognize these molecules. And so what people thought at the National Cancer Institute is maybe HIV has hijacked this signaling system and uses this signaling system as a way to get to the plasma membrane. And they did some really cool experiments to try to test that. So here's a human cell, and the coloring shows that this cell has been modified so that it doesn't have this molecule on the plasma membrane. And there's a cell right next to it that's a normal cell. You can't see it because it has not been modified. So this cell has these PIP molecules on their plasma membrane. So this is a cell where, if this hypothesis is right, the virus should not go to the plasma membrane, whereas in this cell it should. And that's exactly what they saw. So in green now are these viral proteins, and you can see that if there's no PIP in the plasma membrane, the viral proteins stay inside the cell and they don't get where they need to go. But if this molecule is in the plasma membrane, you can see the proteins go there, and the virus can spread. So we thought we'd ask a very simple question using our NMR technique. Is it possible that this targeting could be due to a direct bonding, a sticking together of that matrix domain and these PIP molecules? And so what I'm showing here now, I'm not going to show you a lot of NMR spectra in my talk, but I just want to show you one map just so you'll know what NMR spectra typically look like. These are spots on the map, these peaks correspond to the different amino acids of the protein. And undergraduates in the lab actually assigned these to the different amino acids. And so these numbers refer to the different amino acids on the protein. And if I zoom in on one region, what I can show you is what happened when we just take that protein and then titrate in the PIP molecules. We can see that some of the signals change and some of the signals don't change. And based on the signals that change, we know which amino acids are interacting with the PIP molecule. And based on that kind of data, we could solve the three-dimensional structure or figure out exactly how the PIP molecule is binding. And so here's the first structure of PIP bound to this HIV protein. And here's the ring that has these phosphorus atoms that are shown in red. And there are two tails on the protein. One of the tails is down inside the protein, and one of the tails is actually exposed in a manner that we think might help it interact with a membrane. And one of the most important things as a chemist is we could see which groups are interacting with which other groups. So again, for the chemistry students that have come here, what we could see is that negatively charged regions are interacting with positively charged regions, and it provided an atomic level chemistry understanding of how this cellular molecule is interacting with the HIV protein. And what's really important is that the other different phosphorylated versions of this molecule that are found in other cellular membranes, they don't bind at all. So it completely explains how the virus would get to a membrane that has PIP. But here, I think, is the most interesting thing we discovered. This is a different way of representing the protein. And here's the tail I've been showing you. This is the membrane anchor. And it's stuck down inside the protein. So it won't work when it's stuck down in here. This is the part of the protein where the PIP molecule binds. And what was really interesting, what we found, is that this protein really does work just like a machine. Because when the molecule binds over here, this acts like a little lever. And it pushes on this lever, and it causes this part of the protein to move in. And when it does, it pops out the myristic acid. Now, this was really kind of neat, because now we have a way of explaining how the protein would be attracted to a membrane that has these molecules in it, because it has a high affinity binding site for these molecules. But then this also would provide an additional mechanism to really anchor it tightly to that membrane, because now this can help anchor it to the membrane, and now we have this membrane anchor that's now exposed and ready to interact with the membrane. So here's how we think it binds to a membrane. This is a model of a membrane that somebody else generated, and we're using this to try to model how it interacts. And so this would be like the surface of the cell, the outer membrane of the cell. Here's the PIP molecule in yellow that's interacting with the protein. And then, of course, when it binds, just like I showed you, the myristic acid, the tail of the protein, is now also stuck into the membrane. And we think that this structure now explains exactly how the HIV proteins get to that membrane and how they assemble. But we also think this structure explains how these proteins get to those punctate sites, those very specific sites on the membrane. Now, what I didn't tell you about yet is that these two tails on the PIP molecule are chemically different. One of them has double bonds for the chemists in the audience, and the other one does not have these double bonds, okay? Now, it really doesn't matter what a double bond is. If you don't know what a double bond is, it makes no difference. The point is that one of the tails gets buried and one of the tails is exposed. And the tail that's exposed looks a lot like the myristic acid, the membrane anchor. Now, why is that important? Well, it turns out that these punctate sites where the GAG protein, the HIV protein, assembles, those happen to be very well understood structures on cell membranes called lipid rafts. Okay, it's a very specific structure, and what I'm showing here is how other people have shown that proteins on the cell that normally get into these lipid rafts co-localize with the HIV proteins. It just shows that HIV goes to lipid rafts. And these are cells where this is a different cellular protein that's not in a raft. And you can see that they don't co-localize. And so again, this is just one of the many lines of evidence that the HIV proteins go to these lipid rafts. Well, what is a lipid raft? Well, the membrane of the cell is not homogeneous. It's not uniform. Large parts of the membrane are made up of these molecules that have a funny shape to them, and that's why I'm drawing them in the cartoon form as a bend here. And our PIP molecules, remember, one of them has those double bonds, and so I'm just kind of denoting that by this bend, and one of them doesn't have those double bonds, and so that has this shape. And these kinds of molecules are found in non-raft regions, but in lipid rafts, they're enriched with lipids that have these straight chains, the ones that don't have any double bonds. And so cholesterol also accumulates in rafts. Rafts are really interesting structures in the membrane. Now, cellular proteins that have to get into rafts are often targeted to rafts using two of these saturated chains. Okay? So certain types of proteins in the cell that normally get there do so because they have two chains like this. Now, these PIP molecules, they're not in rafts. So how does HIV get into rafts if the PIP molecule is not in a raft? Well, I can see some of you are nodding because I think you've already figured it out. Here's our HIV protein. Here's the tail, and it's buried, right? So this won't bind to a membrane in this form. Now, if this bumps into a membrane that has PIP, the bent tail gets sequestered inside the protein. And now you have something that has two of these straight tails, and now it looks just like a cellular protein with two straight tails that has to get into a raft. And so now, just with this thing wandering around on the membrane, just like it normally would, when it bumps into a raft, it's going to accumulate in this raft. And so you can see the process of a protein that has to first find the plasma membrane specifically. And once it gets to the plasma membrane, it has to specifically get into a lipid raft. I think we understand that now from a molecular level. It's just the behavior, the chemical behavior of a tail wanting to be buried inside the protein, and then when it gets to a membrane, being pushed out by the structural changes of the lever inside the protein. It's just the same kind of engineering that an engineer would think about when building in a lever. So we can think of these things, these molecules, as little parts of the machine. And then, again, they would accumulate in here because they've decided to try to look like a cellular protein. And we think this actually could be a general paradigm because people have often ask the question, how do cellular proteins get in here? And why is it that in some cells that are dividing, how come those cells have PIP molecules in rafts? And so we think this general mechanism could explain a lot of normal cellular biology that we haven't understood for many years. So I want to tell you a little bit now. So we're hoping that we can use that information to develop a drug. Now, my lab doesn't focus on drug discovery, but if we have an idea, then we will pursue it at least to some level, and then if we can, we'll partner with a drug company. And what I can tell you is that just within the last 2 weeks, we've discovered our very first compounds that bind to that matrix domain, and they seem to block exposure of the myristic acid, even when we add PIP. So PIP still binds, but the myristic acid won't pop out. So we still have a lot of work to do. We have to first see if those have antiviral activity and things like that. So I don't have a big story to tell you about drug discovery on that part, but on a different part of the virus life cycle, we've actually made some progress. We don't have anything in the clinic yet, but I would like to tell you what we've discovered, and so you can get an idea, again, of how chemistry and structural biology can actually lead to a potential new drug. So I told you that once these proteins get to the membrane, they assemble into an immature virus particle. And I also told you that one of the viral proteins called the protease has to cut this protein into pieces, and those pieces then can rearrange to form the infectious virus. And I also told you that the protease inhibitors that block this step have important antiviral activity, and they're part of our drug cocktails. Well, what we were thinking is maybe there's a different way of inhibiting this maturation step that doesn't involve targeting the protease, but maybe would involve a different target. Now you'll notice that these red spheres, this is a protein called the capsid domain. These red spheres go from a structure like this to this coffin-shaped structure in the middle of the virus. And we thought maybe if we understood the structure of this, we could maybe block that rearrangement, this maturation step. And in fact, there have been lots of models of how these proteins have to assemble into this warhead of the virus. So we solved the structure of this protein. This was published in a Science article several years ago now. And so we could see what its shape looked like, and hopefully based on the shape, maybe design a drug. Now, what you can see is that at one part of the protein, there is this structure that's denoted by these arrowheads. This is a beta hairpin. And then I'm showing in a little more detail 2 of the amino acids that make up this protein that interact with each other. And when we published this was really exciting because the virologists had known that in all retroviruses, even evolutionarily very, very old viruses, and viruses, again, that cause leukemia and other human disorders, they all have these 2 amino acids. It's conserved throughout biology. And why do they have those amino acids? Well, what we found is that they stabilize this structure. And because they interact with each other just due to one of them is positively charged and one of them is negatively charged, and those opposite charges attract each other. And so it holds it into the shape. So we thought, aha, maybe you have to have this in order for the virus to be infectious. And virologists showed that that's in fact the case by doing some mutagenesis. So this is an essential interaction, and we thought maybe we could block this interaction. Now, this is the structure of the protein in the mature virus. In the immature virus, it turns out that this is just a different way of showing the same structure I just showed you. This is those 2 arrowheads, only now I'm showing more detail. And this blue and the red are still colored the same here. You can see they're interacting with each other. So this is that part of the protein that I call the beta hairpin. But this is the structure in the mature virus. In the immature virus, before the protease has cut it up, this amino acid is still connected to the matrix domain. Remember, I told you the matrix domain has to get this to the plasma membrane, and once the virus buds, it has to be cleaved here by the protease, and then this rearrangement occurs. And this rearrangement is essential for the function of the virus. And so we thought, wow, let's look at this. This kind of looks like a pocket. And maybe we could design a drug molecule that would bind to this pocket while it's still in the immature form. And then when the protease cuts it, it wouldn't be able to form this because there'd already be something stuck in the pocket, prevent it from doing what it was supposed to do. And it was a graduate student in my lab who had this idea, and I thought it was a great idea. I said, let's go for it. And so he took our structure and used computer modeling to computationally model hundreds of thousands of compounds in the computer into that site. And the computer spit back about 20 compounds that it said would bind much better than the protein itself. So we got really excited about that. We ordered those compounds and tested them. And so here's the other NMR experiment I want to show you. Again, now we're looking at peaks for all of the individual amino acids in the protein. And when we titrate in one of these molecules, you can see most of the signals don't change. And that means that when the molecule binds, it's not changing the structure of the protein. But some signals change. And they change in a very significant way. And based on these kinds of data, we can figure out exactly how tight the molecule is binding and we can even figure out where it's binding. Now, before we tested to try to figure out where it was binding, we also looked at antiviral properties. And this is just a really simple graph. And what it shows is that if you treat cells that are infected with HIV, the cells still produce virus. This means that as we increase the dose of the compound, we're not blocking virus production. But the infectivity of the virus goes way down. And this was our very first lead. And so it's not particularly potent, but it was potent enough to get us excited about it. And so the cell is now producing viruses that are non-infectious. But they don't bind to this site. This came as a huge surprise to us. These green residues are the residues that were affected by these potential drugs. And this is the region where the computers were telling us that the molecule should bind. So this is an example of a fortuitous discovery. It happens all the time. It happened with a certain class of HIV reverse transcriptase inhibitors called non-nucleoside reverse transcriptase inhibitors, where people were looking for something going in one direction and in the process found something that has a completely unexpected effect. Now, it shows you that we still have a long ways to go in terms of developing computational tools to predict binding sites. And that's one of the things that other people are spending a lot of time trying to do. But the good news is now we have something that we know has antiviral activity, and it's binding to this part of the protein in a completely unexpected way, and so we could pursue it. And so I lied to you. Here's one more NMR spectrum. This shows some of the signals. Again, for the NMR people from West Florida, they will know that this shows that part of the drug is interacting with a part of the protein that's deep inside the protein. And this is one way of showing that drug molecules stuck inside the protein. These black lines show us the observable interactions that we can measure in the NMR experiment. And another way of showing that is here. This is a surface of the protein that's shown with this kind of color coding. Here's the part of the drug that's now stuck way up inside the protein in a really unexpected way. Here's kind of a top-down view, so you can see that this really important, what we think is the most important part of the drug, or the potential drug, is deep inside this cavity inside the protein. Now that we know how this molecule binds from experimental data, now we can actually use computers, and they can be very helpful for making something that binds better. And so this is a side view, and what you can see is that inside there's a lot of space inside this cavity. And medicinal chemists would tell you, and they told me, that if we could fill up this space by modifying this lead compound, we might be able to enhance the ability of these compounds to bind more tightly, and they would be better inhibitors of the virus. And so far, we've gotten over 1,000 times improvement in binding just by doing that, by putting on groups that enhance the affinity. So these compounds have been patented, and we've licensed them to companies. The company that we're working with now is in Sweden, and we're just hopeful that we can get them to the point where they can get into the clinic. None of these are in the clinic, but that's what our goal would be. So what I've tried to do is give you a sense for, at least from an academic point of view, how one, as a chemist, would use chemical information and structural information to try to first understand how the virus works, and once we know how it works, to then try to see if there's a way of inhibiting it. And so we've discovered a new antiviral target, small molecules that inhibit capsid assembly by binding to that new site. And based on the structure that we have, we've been able to come up with things that bind much better. If we can get another 2 log units of improvement, we would definitely be in the clinic. And that's what we're hoping to do. Hopefully in the next year or so, we'll make that next big step. And in the first part of my talk, I told you that we think we now understand how the virus knows to get to the right assembly sites inside the cell so that it can bud. And I mentioned that HIV hijacks a normal chemical system inside the cell that the cell normally uses to get cellular proteins to specific sites. And it just hijacks that system. And we think that this can also be a potential new drug target for combating resistance. Now, I want to tell you about the people that did this work. And the structure of the matrix protein was solved by Mike Messiah. He was one of the very first graduate students that joined my lab. He came from Haverford College. And he worked with these undergraduate students. Chiana Pashal was an undergraduate at UMBC in it's what we call the Meyerhoff Scholars Program. It's a program that focuses on supporting underrepresented minorities in the science. I'm going to tell you a little bit about that with the last few minutes of my talk. Mike Messiah actually left my lab. He did a postdoc at Johns Hopkins, took a faculty position at Oklahoma State, got major NIH funding, got tenure. Was the only black faculty member I think, in the entire college. He very recently moved back, and he now has a faculty position at George Washington University. Mary Starich ended up going to the NIH, where she's a staff scientist, and Chiana Pashal was an undergraduate. She ended up going to UPenn, getting her MD, PhD. Actually, UPenn has the oldest medical school in the United States, and yet the first African-American MD, PhD that in the history of this school that graduated from UPenn was one of our Meyerhoff scholars. Now, the work that I talked about was led by this group. Chun Tang was a graduate student in the laboratory at the time. He actually took a faculty position at Missouri, and he was only there for a couple of years before he was recruited back to China. Now, he's at Wuhan University. They gave him a huge amount of money, and he's a full professor at a very young age, so it was really good for him. He worked with these undergraduates. Now, Isaac went to the MD PhD program at Johns Hopkins, and he's finishing up now. But I really want to tell you about Erin Loliger. Erin was homeschooled in Maryland, and she started taking college courses when she was 12. She enrolled full-time at UMBC when she was 14, joined my lab when she was 14. She had her first publication, The Proceedings of the National Academy of Science, when she was 16. She had her second publication when she was a senior at 18. And when she was 18, she was leading this group of undergraduates who she was mentoring herself. Erin, actually, when she left UMBC, she joined the MD PhD program at Harvard, and that's where she is now. And the students that she was mentoring joined the MD PhD program at Maryland. The PIP project that I told you about was led by Jamil Saad. He was a postdoc in the lab working with these well, okay, they're not all undergraduates, but working with this group of students. And Jamil is now an assistant professor at the University of Alabama in the medical school. The work on the capsid inhibitors was led by Samson Sheree. He was a Meyerhoff undergraduate scholar who went to Maryland to do his MD PhD, and so he did his PhD work in my lab, and he's now finished his MD and the PhD, and he's now a resident. Yasmi Nadassa was an undergraduate in the lab, and she went on to join the MD PhD program at Harvard. And then Peter McCready is a current graduate student who is working on the matrix inhibitors that I just briefly mentioned at the end of my talk. And this is a picture of my lab from last summer, and you can see it's a pretty big group. I have usually I'll have about 6 graduate students and a couple of postdocs, but I work with a large number of undergraduates, and that's because I was inspired by the work here that I got to do as an undergraduate at West Florida. So not only do we have undergraduates that work with me during the academic year, but in the summer we have outreach programs that bring students from all over the country who come and do research just for the summer. And so this includes that larger number of undergraduates as well. So with the past few minutes, I don't see a clock in the room. So that's good for the speaker, but bad for the audience. What time is it, Mike? It's 7 o'clock now, so I'm over time. I'm going to just very quickly tell you about what I think is the importance of undergraduate education. In my lab, I've had almost 200 undergraduates since I started working with them. About 65% are women and 45% are from underrepresented minority groups, and that's groups. Both of those are groups that you don't see generally attaining high-level positions in academic careers. And so that's our goal, is to try to get more people in these groups in high-level positions. And so this was just one group of seniors that were working in my lab that graduated 1 year. So Chelsea, MD, PhD at Penn, Wash U, MD, PhD at Yale. And this group of undergraduates had 12 papers. One was in science, and 3 of them were first authors, and 3 of them got their papers, got the covers of the journal. So these are really smart students. And most places, most colleagues, when I go and talk about our diversity programs, they don't believe that we have these large numbers. African Americans, they only make up 12% of enrolled college students, but they make up only 2% or about 3% of science and engineering PhDs in the US. And science retention is a general problem, not just among minorities and women. Research and development depends really heavily on foreign students. I don't know how many of you knew this, but in the United States, 65% of the PhDs in engineering go to international students. Now, I think it's good to train international students, but why are so few US students staying in the sciences? Why is it that half of our students who we educate are going into other fields? And so part of it may be jobs, but there are certainly jobs for the international students. And the international students, by and large, are staying in the US. So why should we care about diversity? There's a need for minority leadership, obviously, a need for a minority workforce. Health disparities is an area that I care about. If you have African American friends, then you probably know that your black male friends are 350% more likely to die of high blood pressure than you are. 1 out of 4 African American women over age 55 has diabetes. Lupus is a huge problem. So there are clear health disparities that could be addressed better if more minorities were educated about these. And then we also have to care about who's going to be paying into my Social Security when I'm ready to retire, because if you look at US population projections, and this is from the Census Bureau, by 2030 or by 2050 now, Caucasians will make up less than half the US population. So we've got to think about who we're educating. And this is the president of UMBC. He's just a phenomenal guy. He's been the president for almost 20 years. His name is Freeman Hrabowski. He was raised in Birmingham. One of the girls that was killed in the baptistry church bombing was a friend of his. And so he was actually incarcerated at 12 years old for leading a youth march. But his parents cared about education. They were teachers. And so he's been the president. He started this program that focuses on diversity and retaining minority scientists. And it has elements that work at lots of places, a summer bridge and mentoring and research experience. And it's not just a minority program. More than half of the students are African American, but there are still a lot of Caucasians and Asian students that care about issues of diversity. And this is what I think will impress you the most. We typically get about 2,000 nominations a year mainly from Maryland teachers and counselors who have students that they think would be qualified for our merit-based program. It's not a remediation program. We're not looking for kids who come from tough backgrounds. We're looking for educated kids who have had AP calculus, and we're getting this many nominations. We typically get over 700 applications. 850 students have been supported, and about 90% of the 600 graduates who have gone through our program now have earned science degrees. So these are high achievers. It was started by Baltimore philanthropist Robert Meyerhoff. We've had 74 of our alumni have now earned PhDs. So our program is 20 years old. We've had 150 have earned master's degrees, 115, about 80 have earned MDs, and 300 are enrolled in graduate or professional school. These are huge numbers for minority students. We're the top producer of black students who now go on to PhD programs and MD PhD programs in the US. And so, this is one group in a program that I have a grant from Howard Hughes that support. You can see these are 4.0 students. This was the second graduating class. Now, who wouldn't want to work with these students? This is just a group shot that I took a while ago, and the programs that they matriculated to, Hopkins, Stanford, Florida, Baylor, These are the brightest of the brightest. So we think we've developed a successful model for attracting and retaining talented minority students, and what we're now doing is applying this to all students. And it actually works. What works well for minorities and what works well for women works well for Caucasian students too, which is not a big surprise. And we think that these programs that we're developing could help address the national needs if more broadly applied. And so with that, I thank you for bearing with me, because I did want to talk a little bit about our pedagogical efforts, just because I think they're so important and because I think that it's something that William Smart would have appreciated, and I think Mary does as well. Thank you. Yes, the mic, well, he was faster. 1 second.
Hello?
Yes.
Dr. Summers, you're a miner. You mine chemistry and imaging technology to bring these brilliant breakthroughs, and you also mine the people of the world to find the best who might otherwise slip between the cracks. So for that, you have my congratulations.
Thank you. When you said I was a miner,
you're not that young.
I don't want to tell you what I thought. I was criticized my whole life for looking a lot younger. I'm 53, and I was been criticized my whole life for looking too young. So, sorry, go ahead.
I have two quick questions. One of them is, when you're looking at candidate compounds to fill particular needs to inhibit the life cycle of the virus, do you get any advance indication of what toxicity might come along with those compounds? And the second question is, even in the face of all the brilliant breakthroughs that you're making in understanding how to inhibit this virus, is anybody addressing the ongoing problem that promises to undermine those efforts in people not being compliant about taking their medicines, and then we'll just be in round 3 of the same fight?
Right. So I think that those are both really good points. So the first point is that I was not trained as a medicinal chemist. But these days, there are actually significant numbers of young people who are getting into college, who are going into chemistry programs, who are interested in forensics and medicinal chemistry. And I think that's one of the things where the popular TV shows have really helped us. They watch CSI and think that looks like it would be fun. And so, so there are people now, larger numbers, who are being trained who could look at a molecule, the one that I put up there, and would say, well, it's a nice lead, but it will never be a drug because it has this kind of atom on it in this spot, and we know from experience that that won't work. And so what I have to do is I have to partner with people, and that's why I have partners with people in Sweden who have the expertise in drug discovery and actually have the resources to make lots of compounds, and we do things as a collaboration. More and more these days, people really have to collaborate in order to take big steps forward. And so it's exciting to make that initial discovery, to have an idea, and to get that initial lead, but I understand that there's a limit to what I can do. Your second question is, one that's more of a political nature in a way. What do you do about people who are not compliant? And it's a bigger problem than just compliance, because even in the US, the drugs are expensive. I just learned today, here this evening, that Florida has the second, if you look at all the states in the US, Florida is number 2 in terms of the number of HIV positive people living in the state. And I know from articles I've read and physicians I've spoken with that when people are first diagnosed with HIV, they're not put on drugs right away, but people wait a period of time until they actually start taking the drugs because of the costs associated with them, and also when is it really beneficial? Well, now there's pretty good evidence that the sooner you start hitting people, the better. In fact, the drugs can even be taken prophylactically in people who are high risk. And so, to benefit. And so the real question becomes one of cost versus benefit. And there's always a cost-benefit ratio. And so people will have to figure that out. Now, there are communities in the U.S. who are very well educated and very well organized, and people who are educated and have the resources, it won't matter if you get the message to them because they've gotten the message. But again, there are people in society who may get the message or may not, but because of circumstances, may not be compliant. And so it's those individuals that really generate the greatest risk, the people who are in the sex trade. And you can't just go out and say, it's bad to be a prostitute, don't do it. Or you can't just say it's bad to have multiple partners, don't do it. Or you have to stay on your drugs no matter what other sacrifices you have to make. There are certain people in certain conditions where that just won't happen. If you're an alcoholic, you don't even have to be HIV positive. You may know people who are alcoholics. People deal with different problems. And so education, we've got to keep education high on the list, but it probably won't always be enough. You were next.
Yes, I also graduated from UWF in 1980 with a master's in management. So our paths didn't cross, but it prepared me for a good career for my future. And I want to say I hope you had as much fun as I did while I attended the university.
Well, I'm not going to talk about the fun that I had. But it certainly brings back, I can't talk about all the fun. My wife came with me this time. I met her shortly after when we were I was in graduate school and she was an undergraduate at Emory, and that's where we first met. But it's interesting because, to repay me for tutoring her, I met her because she needed help in PChem. And so I was tutoring her, and she said, I'd like to repay you for tutoring, can I take you to dinner? She said, any place you want to go. And I said, any place? I said, I have a favorite restaurant. So she said, yeah, any place. I said, okay, I'll pick you up Friday, bring some clothes. So we came back to Pensacola to Marcello's. And I was so disappointed to hear that Marcello's is closed now.
Yeah. Well, I stayed in Pensacola. I didn't go to Harvard or any of these places.
I'm envious. I love coming back here.
Yeah, I hope I didn't sound critical or sarcastic when I mentioned jobs or the economics, but I raised a very important question from the research you're doing. I'm a physician. The healthcare profession, as well as the space profession, have had, as you know, a lot of growth in the technology and the developments. Unfortunately, those accomplishments actually surrender to the politics and the economics that fund them. We're going through a major challenge in medicine that we're being essentially criticized that we use technology too much and that the cost of that technology is too expensive. The space program, where we have some of our best engineers in the country that have some of our best scientists, have the same problem. The incentive is not there because the politics and the economics actually suppress it. From your standpoint, though, in research, do you have people that actually work with the economical development of your products or your drugs so that you can make it cost effective, or to actually mitigate between the academic institutions that develop the accomplishment and the field where the medications or accomplishments are going to be applied, so that you can have a bridging of the gap between the politicians and the funders and the people that work and do the research to develop it?
Right. So there's not an easy answer to that problem. Politics is going to play an important role. There's always going to be tension, and it's not just the academic community versus the scientists, or government versus the drug industry, or the lobbies for Medicare and Medicaid versus whoever else. Even within the scientific community, we have these kinds of debates. What is appropriate? There are certainly diseases that affect huge numbers of people worldwide that no drug company is trying to go after because they affect people in third world countries who can't pay for them. So should the NIH spend money on drug trials that companies won't do? If you're a humanitarian, you would say yes. If you are practical and you look at the financial situation in the country, you'd say no. And so there's going to be tension. And I think that's just common. And so in terms of the second part of your question, and I think that's why we have to continue to have debate. And that's why it's important to have these discussions in the public and to get different points of view. And I don't have the answer. I have my perspectives, but I don't have the answer to that because the answers always cost money. The second part of your point is, are there mechanisms to try to help people in academics get their compounds to the market in an efficient way? And really, the governments in the states try to push that. I mean, one of the reasons they want an academic institution here in Pensacola, and a good academic institution, is because companies might spin off. Or you might attract more business here. Business likes to go where smart people are being produced and educated people are being produced. And so, you know, the more jobs, the more tax income, you know, the more prosperous places are. So in Maryland, we have what's called the Maryland Industrial Partnership, where the government will actually provide matching money to faculty members and people in industry if they partner up and try to do things that could lead to a new drug. We have a whole Maryland biotech industry that has been set up where there are institutions around the state to try to spin off companies. And so different places have taken different approaches, but it's not a perfect system. So in my case, I would like to have more people around who could give me better business advice. And again, if you're at MIT, you're going to have really smart people, you know, that can't wait to help you get your product to market. If you're at a smaller place like where I am, you know, it just depends on how overloaded the offices are.
Thank you for time.
Yes.
One more question.
Thank you for a wonderfully engaging message, and thank you for your passion for young people and education. Your model, most medical models are Newtonian in that they're mechanical, looking for the mechanical edge. Years ago, there was a man named Royal Raymond Rife who allegedly developed a microscope that allowed him to see the first living virus. And he thought Einstein had the right approach, and that everything was all about energy. And he and his colleagues, they discovered what they called the resonant frequency of maybe the cancer virus. And they were successful in killing cancer viruses with sound, with RF energy. Does anybody today have any interest in doing, I guess, medical treatment or research with energy or electricity or along that line?
Right. So I think that those kinds of things are being done. It's more outside of the kind of work that I'm doing, but trying to use different sorts of electromagnetic radiation, just normal light that can penetrate certain depths under the skin and then be focused in a way that could target certain cancer cells. I know that people are doing that kind of work. And I mean, the cancer field, you know, I think cancer would be solved by now if there was only one kind of cancer. But there are so many different causes and so many different ways that it spreads. And so I think that that's just a bigger problem. I mean, certainly certain kinds of cancers, if you get testicular cancer, if cancers are caught early enough, then a lot of the normal treatments really are very effective. But in terms of using energy like UV visible or even sound, I think ultrasound is used not to treat cancers that I know of, but to treat other ailments, break up stones and things like that, because it can be focused. And I don't know, it's a little too outside my field to know if it's, yeah.
It's time for our speaker.